Cape Nature is the provincial conservation agency in the Western Cape. This project is part of the Cape Nature's Invasive Species Management Unit project, and the aim of this project was to eradicate smallmouth bass from the Rondegat River. There are many threats to our freshwater native fish in the Western Cape, but the exotic invasive alien fish that was brought in for angling species like the smallmouth bass are regarded as the highest threat for these fish. The highest concentration of threatened freshwater fish in South Africa is in the Olifantsdoring River system, and the Rondegat River is a tributary in this system. The purpose of the Rondegat project was to eradicate invasive alien smallmouth bass from 4 km stretch of the lower river, and also to eradicate the alien plants in the riparian zone. There have been critics of this project because we used rotennin, and rotennin is there to kill the invasive fish, but it also has an effect on the aquatic invertebrates. The monitoring of this project is a critical component of the project because that will inform ourselves and other stakeholders of the success of the project. This will also guide us in future projects where we use rotennin.
We have just completed the first year of what is a 3-year funded project being run by the South African Institute for Aquatic Biodiversity in collaboration with Rhodes University and Stellenbosch University. The objective of the invertebrate component of this monitoring program is to look at the insect community here in the river and assess both what the impacts of the rotennin was on the community as well as to monitor their recovery over time. So a critical aspect of this monitoring program here on the Rondegat River is that it has been independent of Cape Nature, which follows international best practice. And the Water Research Commission has funded the work and have been very helpful in that regard. So we assess the invertebrate community using the South African scoring system, which is a rapid bioassessment technique that gives a fairly broad impression of the ecological integrity of the stream. We also collect individual stones from the stream in order to get a quantitative assessment of invertebrate abundance, which is standardized by the surface area of the stones. As part of the SAS monitoring technique, we also collect water quality data such as pH, conductivity, and oxygen content. During the first rotennin treatment, we saw certain very sensitive groups of insects were negatively affected, particularly the mayflies. So while the analysis of the invertebrate diversity is still ongoing, what we can tell from the preliminary biomonitoring data is that the community has been fairly resilient to the rotennin treatment, and that the community is continuing to recover well in the river. Our preliminary data also indicate that since the removal of invasive wattles and gums upstream of the rotennin treatment area, the insect communities have also started to recover there quite rapidly. Some of our results have already been published in the international literature, as well as in a Water Research Commission report which can be found online.
This has been a wonderful teaching opportunity for the students from our department. We've had a number of MSc students and PhD students involved in this program, and this is now the 3rd year that I've brought our honours class up to the Rondegat, and we use them as much needed manpower, but it's also a teaching and learning facility for them. We encourage our students to be independent thinkers and to really think critically. So when we tell them we're coming up to a sampling site where we've poisoned 4 kilometers of river, we arrive here with a mixed bag of feelings. We start off by showing the students the bass zone. They snorkel in the river, they have a look and see what's there, and to be honest, there's pretty much nothing there. After a few minutes in the water, they see a couple of bass. if they're lucky, they see a few insects. This is pretty much what you'd see if you looked in just about any river in South Africa. We then move up to the pristine area above the treatment site, where bass have never been introduced. And they are blown away every single year. For the first time, they put their face into a body of water, and it's teeming with life, with catlets, yellows, red fins. We then work down the river system through the treatment zone, and only after a year since the rotononing, the students see firsthand the impact that the rehabilitation has had in such a short amount of time.
$130. Zero Barbara's calendars.
So in the treatment zone, they're now seeing hundreds of little indigenous fish that originally weren't there.
So as part of my MSc project at Stellenbosch University, I'm looking at the effect of the rotenone treatment on the Rondegat River and on the fish communities within the treatment zone of the river. The reason why I'm doing it is to determine whether this rotenone treatment has been successful in removing bass and allowing the recovery of native fish within the treatment zone of the river. The way I'm doing that is I'm using cameras to film different sites, both up at the top in Algeria and where the river is pristine, down here in the control zone, and also below the waterfall in the treatment zone. We use underwater footage, we snorkel all the sites, we electrofish the sites, and we also seine net the sites to get an idea of what's going on in those pools and what the fish are busy doing. What we found so far is that no bass have come back into the treatment site since the second rotenone treatment, and there's also been a good recovery of native fish, mostly juveniles, within the treatment zone. This is a Clanwilliam rock catfish, a threatened endemic species only found in the Doring olifant system.